What I'll be doing is helping you to get the resource book into the paper, right? So your A-level bio, the trend that is going on is that you have to get the resource book into the paper somehow, right? So through you, the resource book is going to go into the paper. So that is the trend that is happening. It's a bit of a sad tendency, but we can't do anything. So what I'm going to help you is through me, right? I'm going to be sort of on the side here. So I'm going to be helping you get the sort of resource book into yourself, all right? Through helping you understand, helping you memorize, and helping you practice the concepts in the resource book. I'll be helping you to get the resource book into the paper, all right? So as I was saying, I hope by now you have a good copy of the resource book at hand, and I want you to be prepared with it before we start the class. Because as you remember, in the first tute into the introduction to bio and also the cellular basis, I gave you the resource book content in the tute. So I won't be giving you the resource book content in the tute anymore, right? This will be just a few diagrams. Your tute will be just some diagrams and the questions. That will be it, right? I won't be giving you anything from the resource book in the tute, right? You have to have the resource book separately. The tute will be just a small note and a bit of some diagrams that will help you to understand the concepts better. That will be it. Okay. So, am I clear? Any questions? Any questions that you want to ask before we move on? Yes or no? Any new students that have joined today? Do you have any doubts, Puta? Right. So, we will get into the lesson that is from page number 24 onwards. So, the commonest word that you're going to hear for the next maybe 3 weeks, we'll be able to finish this within 3 weeks. This section that is cellular basis is going to be the word cell. Cell, 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 cell. Okay. So, this word cell has a historical background, right? You have to know a bit of a history, right? So a cell is can mean many things, right? There are prison cells, there are cells in a beehive, right? There are battery cells. I mean, you can use the word cell to mean any sort of compartment, right? Any sort of compartment, right? So that's the inspiration. That was the inspiration. That's what gave the idea to the person who gave the word cell to the smallest unit that we can call as alive, right? By now you know the organizational levels, right? What are the organizational levels of life? We usually start from where, Puta? I mean, where do we usually start our organizational levels of life? I'm going to start. Do you know? So what's CJ? That's his first name. Uh, so so I sort of am a bit reluctant to start there because of that combined maths reputation. So I'll be starting at a different institute called Royal Institute. I mean Royal. It's just the name in Nugegoda, not probably at CJ. Right? What's his name? Like I don't remember. So yeah, not next week, but probably the week after next. I'll be updating you on that. Right? So if you are able to come to the physical class, then you can. So as I was saying, uh, Order and organization is a basic living characteristic, right? Order and organization. So we usually in bio, we don't go into much depth. There are subatomic particles, then there are atoms and things like that. But we just start with molecules. Molecules is fine, right? Biological molecules. So we talked about biomolecules now. We divide it into four main groups as carbohydrates, lipids, proteins, nucleic acids. So these molecules will organize into organelles, right? Organelles, or actually organelles or subcellular components. Subcellular components, right? Subcellular components. Then what is the next step? We are finally arriving at the level that we can call as a living organism, that is cell, right? So we talked about the molecules. Now we are going to talk about the subcellular particles in the next step, because in the end, what are we going to study? We're going to study cells, right? So, uh, we usually describe cells as the basic structural and functional unit of life, right? Basic structural and functional unit of life. I think you know about this from O level as well. Structural. So this applies only to what sort of animals? What we call as multicellular animals, animals who have more than one cell, right? So if you take us, we are composed of trillions of cells, right? And if we break down us into the cellular level, you can see we as a human is composed of these trillions of cells, and our building block is a single cell, right? Different kinds of cells get together. As you build a house with the individual bricks, we are composed of the individual cells, right? And functional, right? The cell is able to perform all the living functions of a organism, right? What are the things you know? Respiration, right? It can produce energy, right? It can produce energy from other chemicals, or it can produce chemicals that are very energy dense, what we call as energy molecules, by a process called photosynthesis. Then it can make other things, like it can make proteins, can make carbohydrates, it can copy DNA, right? It can do so many things, right? So therefore, the cell is the structural and functional unit of life. Any questions? So if we go to the resource, they say all organisms are composed of cells. The basic unit which can be called living is the cell, which may be a single cell organism or a multicellular organism, right? Multicellular plant or animal. The level of organization of matter represented by a cell shows all the characteristics of life. Any stage below the level of a cell cannot be considered living, whether it's a single celled organism or multicellular plant or animal. So that you know, right? So we don't consider these subcellular components, organelles, or molecules as living, right? So the history. So as I said, the person who gave the name cell, what's his name, Puta? He's called Robert Hooke. In 1665, he looked at a cork. What is a cork? That is what we, in singular, we use Kirala plant for corks. There are other types of sort of plants that we can use. Yes, that we can use as corks, but in Sinhala, in these sorts of Ayurvedic medications, and also in our old Araku bottles, we used to use Kerala as the cork, right? The stopper. So he examined a thin slice of cork under a microscope that he sort of invented. It is not a simple microscope, as they say, right? Uh, and he gave the term cell. So if you go to the tube puta, you have a diagram here, right? Do I need to screen share? Okay, I will screen share. right? So this is going to be the trend that will be happening with the tute this point onwards. I want you to write a few things in the tute, right? Write down a few points, nothing much, a few notes that I give you. You need to write down in the tute, just some additional information. So you can see my screen, right? So uh, this is the Robert Hooke's microscope that we consider. This is, I think, a remake of it, not the original one. This is the microscope he used, and this is the view that they say he observed. Right? Even at present, you can observe this. Right? So a thin slice of cork, and he observed it under this microscope, and he could see these individual compartments. Can you see? They look like. Have you seen a beehive? Right, in the honeycomb, in the beehives, Mivade, you can see the individual compartments. So they look like those, right? So that's why he gave the term cell. So please write down here in this space that I have given. If you don't have the tube, please write down on a bit of extra paper and then take it to the tube when you have the printout, right? Robert Hooke, Robert Hooke introduced the term cell, introduce the term cell, introduce the term cell to describe the basic compartments, to describe the basic compartments to describe the basic compartments, to describe the basic compartments he saw under the microscope. To describe the basic compartments he saw under the microscope. Give me 1 minute. Okay, give me 1 minute. Okay, sorry. All right. Then you can label this diagram. You can label this diagram as the cork cells that he observed. Cork cells that he observed. Okay. Please let me know if you have any issues seeing the board or the diagrams. All right. So when I screen share, don't you have a camera view of me? You do, right? You do, right? Yeah, yeah. So not just the screen, right? Yeah, okay. Yeah. Okay. All right. So then we can continue. So what is then the next person that you have to know, Anton van Leeuwenhoek, a contemporary of Robert Hooke, was the first to describe and record living organisms, euglena and bacteria. Right? So contemporary means what? That means at the same time, right? So as you can see, he was living in 16. Robert Hooke was 1665. Leeuwenhoek was 1650. So he was observing this at the same time as, I mean, in the same sort of, let's say, same time period as Robert Hooke. He didn't give the word cell, but he was observing. He was observing these sort of unicellular organisms, such as euglena and bacteria. Right? Euglena, you can write down uh, by drawing an arrow. Euglena is what we call as a protist. Protist. Euglena is a protist. Euglena. It's not a bacteria, right? But it's single cell, unicellular, and we cannot observe with the naked eye, right? Euglena. Then we come to three people that we together consider as the fathers of the cell theory. Okay, fathers of the cell theory. Matthias Sladen, Theodor Schwann, and Rudolf Virchow. These three people we consider as uh, the fathers of the cell theory, right? Cell theory. So Matthias Sladen was a botanist, right? Botanist means he's mainly concerned, his field of study was plants, right? So he concluded that all plants are made of cells, right? A plant is what, Puta? It's a multicellular organism. No? So if you take even a very tiny plant, it has millions of cells. So he observed such a cells, such a plant tissue, maybe leaves, roots, whatever, and he concluded that plants are composed of these cells, right? In 1831. Then Theodor Schwann, he was a zoologist. So his study was, I mean, studying of animals. So you know, what are the three uh, main branches of biology? Zoology, botany, and microbiology, right? So these two were studying two of the main branches, that is botany and zoology. Then finally, Rudolf Virchow in 1855 showed that all cells arise from pre-existing cells by cell division. Right? What is cell division? Have you seen cell division happening? Did you learn that for levels? You do, right? Mitosis, meiosis, you have, no? So you will learn. We'll have to learn about mitosis and meiosis in detail. So this is an animation of how cell division takes place. We'll have to learn about all these things that are happening in this animation. This is cell division. Okay. This view is better, no? Yeah. So one cell is going to be 2. So Rudolf Virchow saw this cell division happening in bacterial cells, right? They saw bacterial cells dividing, and he understood that the cells don't spontaneously uh, occur. They are right. I mean, they don't automatically form. Always, before one cell, there was another cell that gave birth to that cell, right? All cells came from a cell before. Right? So you will ask what created the first cell, right? That we learn in the third lesson, right? There's a theory that I told you this, right? Did I tell you about how the first cells originated on Earth? Did I? No, right? So that we have to learn about when we are talking about our the very first Earth conditions and how the life originated on Earth. So that is a different lesson. That is our third lesson called the evolution and diversity. So anyway, this Rudolf Virchow said that all cells arise from pre-existing cells, called by a process called cell division. All right. So together, these three got together and presented what we call as the modern cell theory. Right? Modern cell theory. So please write down the modern cell theory here. In the space, I think you have some space here, right? In this space, all right? Cell theory, small topic, and put 1, 2, 3. These 3 points you have to know by heart, right? What are the three things? All organisms are composed of one or more cells. Number two, the basic structural and functional unit of life is the cell. And number three, all cells arise from pre-existing cells. This is the cell theory. Okay, this is the cell theory. You have to know these three by heart. Very commonly questioned in the structured essay paper. They will directly ask, what are the three principles of the cell theory? So please write down. Number one, all organisms are composed of one or more cells. All organisms are composed of one or more cells. The basic structure and functional unit of life is the cell. Number three, all cells arise from pre-existing cells. So you have to know 5 people, that is Robert Hooke, Anton van Leeuwenhoek, Matthias Leiden, Theodore Schwann, Rudolf Virchow. 5 people enough for the history of the cell. Right? There are other people like your, let's say, your Ernest Haeckel and your Singer Nicholson. There are so many other people that we'll sort of encounter in the future. So not right now necessary. Okay. Any questions? Now, I want to tell you a bit about the diversity of the cell, right? The diversity of the cell. So you can see 3 diagrams on the very top of the tube. 3 cells. Anybody know what are these 3 cells? Anybody know? This cell, and this cell. Those are 3 types of cells that we have in our body. Anybody know? What are those cells? Yeah, very good. I got one answer. So this is, I just put those diagrams so you can see how much of a diversity, even within a single organism. So this has to be a multicellular organism, right? Composed of more than one cell. If you take a complex animal like us, there's such a diversity of cells, right? So many different cells, right? So yes, so I got correct answer. So please name these cells. So this is the sperm, right? That is the male gamete. I'll tell you, right? Sperm, and this is a egg, right? Egg, or we can call it as the ovum, and this is a neuron, right? Or we can call it as a nerve cell, right? So I just put those to give you an idea about the diversity, as I said. So write down under the sperm as something a little for your interest, right? Write down under this. Write down under this. Smallest cell in the body. smallest cell in the human body. Smallest cell in the human body. Smallest cell in the human body contains smallest cell in the human body contains paternal. Paternal. What is paternal? That means your fathers. Contains paternal genetic information. Contains paternal genetic information. You will have to study the structure of the sperm, label the parts, and describe each of their functions when we are doing reproduction. So don't think this is out of syllabus. No. You have to know. I mean, when we're doing these early fundamental lessons, you have to have an idea of the basics as well. All right? Basic things. So what did I say? Contains paternal genetic information. Then under the second one, this is your egg. Write down the largest cell in the human body. The largest cell in the human body. Yeah. Largest cell in the human body contains the maternal. Maternal is the opposite of paternal. Maternal, mothers. Maternal genetic information. Maternal genetic information. Right? The third one, neuron or a nerve cell. There's not much space. You can write down here maybe in this space here. Neuron. The longest cells in the human body. Longest cells in the human body. Longest cells in the human body. They are not able to divide. They are not able to divide. They are not able to divide. Longest cells are your neurons, and they are not able to divide. So when you are born as a baby, the number of neurons that you have when you are born and at the present, and when you die, not when you die, okay? So at the present also, you might have killed off some of your neurons, right? But when you are born, you have your maximum number of neurons, all right? So when you get older, especially after you are, when you get something like a non-communicable disease, or if you are an alcoholic, or if you have other unhealthy lifestyle patterns, your neurons can be damaged, right? Whether it's in the brain, whether it's in the spinal cord, right? So neurons, the number you get from your birth will be for life. They won't be regenerated. For example, if you cut the skin of your finger, a new skin will originate, right? A new skin will originate. If you cut your hair, the hair will grow back. But the neurons are not able to do that, right? Because it is not able to divide. And also, what did I mean by when we are saying they are the longest cells? So I show you. Did I have a microscopic image for you? So a neuron. So there are other sort of supporting cells for a neuron. A neuron is this. This one is a neuron, right? sorry, this yellow one is the neuron that is sort of in the center. I hope you can see. And can you see how long it is? So this, the entire thing is the neuron. The other this blue color, purple color cells they are just supporting cells. This is the neuron. They are very long, right? And also if you take, so this is our spinal cord, right? So this is like a cut part of your spinal cord that is running through your backbone, the back, right? That's spinal cord. So one end of a neuron can be located in your spinal cord and the other end can run through entire length of your hand until it reaches your fingertips, right? That is one entire neuron. But hundreds and thousands of those neurons get together to form what we call as a nerve. Snayu කියලා කියනවා, right? Snayu. Have you ever touched a nerve, right? You can use the different terms to say you are getting on someone's nerve, you touched a nerve. But have you ever actually touched a nerve? අතින් අල්ලලා තියෙනවද nerve එක? Do you know where we can touch a nerve? Anyone? Brain cells can't be damaged due to high screen time. No, maybe your retina, the cells in your eyes, they are not neurons, right? They have a pigment, right? We'll talk about that. So as I was saying, have you ever touched a nerve? I'm giving you a hint right now. Have you touched a nerve in your body? Can you see my camera feed? Yeah, right? Where? Where can you touch a nerve? Current passing? Yes. It feels like a current is going through your hand. I'm giving you a hint. Have you ever bumped this place on your elbow somewhere to a door or whatever, to a sharp edge? Desk එක වගේ වැදිලා තියෙනවද මෙන්න මෙතන? In your elbow. There's a nerve passing there that is very close to the skin. Called the ulnar nerve, right? Ulnar nerve. It's a bundle of neurons, right? Have you ever bumped here? Then your entire hand, especially where? This side, no? The inner side of the palm gets like getting pins, you're sticking needles into it, right? That is because that nerve is running from your sort of spinal cord that is in your neck and also in your upper back through the arm, through here, and it is supplying this part of your arm, the inner part, right? So that is a very good example for a nerve that is very close to the skin. Otherwise, all the other nerves are very deep in your body, right? You can't touch them. Okay, so that is just an example. So as I was saying, there's such a big diversity of neurons, and so a big diversity of cells, and we are going to explore what is so, let's say, what is so special and what gives these cells such special characteristics. How are they able to be considered as living things? All right. Any questions up till now? Any questions? So, if not, we can move on. So, there's a sort of a chart in your page 25 that we are comparing our prokaryote and eukaryote cells, right? So, what I am going to do is skip it for now. We are going to come back to it. And if I forget, please remind. So in page 25 and 26, you have a chart. Right? Page 25 and 26, you have a chart. I think one of the mistakes the people do when they're teaching this is attempting to teach this chart before completing the rest. Right? So what I'm going to do is, we are going to skip this chart in page 25 and 26, and we are going to move on to page 27 in the resource book. Hands up, anyone who hasn't got the resource book? Do you have the resource book, Putha? Anyone who hasn't got the resource book? Do I need to screen share? Anyone? No, right? Everyone has the resource book, right? It's fine if you don't. All good? I hope so. Okay, right. Then we can move on to study our first, let's say, the structure of the cell that we can study. So before that, I'll screen share a bit and just give you a tour of the cell, right? Just a tour of the cell. Before we get into the small aspects, just have a look from the outside using the software. Okay. So we can, I mean, I'll show you a bacterial cell as well. Just let's take a look at this animal cell. So this is, I hope you can see the screen. Can you see the screen? Yeah, right. Yeah. So this is sort of been cut open, the cell. So this outer covering, I'll just give you the names. No need to worry. I'll be teaching every part, right? So this outermost covering is what it's called the cell membrane or the plasma membrane. Cell membrane or the plasma membrane, right? And inside the cell, other than these different colorful structures you can see, it is completely filled with a gel, right? It's a jelly-like substance that we call as the cytosol, right? It's a jelly-like, mainly water-based, right? It's a semi-fluid, right? Jelly-like substances that we call as the cytosol, right? Otherwise, all these colorful components are sort of immersed in a jelly, right? Imagine you are making a jelly with different things in it. You are putting cashews and what else? So imagine a jelly, right? So a cell is like that, surrounded by a covering that is called the plasma membrane or the cell membrane, and all the things inside the cell are sort of suspended in that jelly that we call as cytosol. All right. Then I'll just give you some. I'll remove this part of the plasma membrane as well. So this I can remove the jelly as well. Now you can see the biggest structure in a animal cell is sort of inside here, right? I'll remove this sort of structures. Now you can see this purple colored the dome, right? It's a massive sort of a structure in the cell that we call as what? Any guesses? What is this structure? Any guesses? The big purple colored one. Any guesses? That is the biggest structure that you can usually see in animal cell. Anyone? Not the nucleolus exactly. I hope you're trying to say nucleus. Nucleolus, no. This is a nucleus. I'll show you a nucleolus. You're not incorrect. I mean, so this is a nucleus, right? Nucleus has its own membrane. It has its own covering, and we'll remove that covering. It is called the nuclear membrane. Now you can see the inside of the nucleus. This is the nucleolus. I'll explain. It is sort of a dense area inside the nucleus that we'll talk about. Inside the nucleus, you have the DNA, right? These are DNA. This is not entirely accurate. DNA is not like this in a normal cell. But for you to understand clearly, the DNA has been indicated like this as chromosomes. Right? Then what is this light blue color? That is called the rough endoplasmic reticulum. It's a network of membranes that is surrounding the nucleus. I hope you know these structures. You know from O levels, right? Rough endoplasmic reticulum. It has these small subcellular components that you can see. Can you see the small dots? These are called ribosomes. These are involved in the making of proteins, protein synthesis. Then outside the rough endoplasmic reticulum, we have the smooth endoplasmic reticulum. They're sort of tube-like, right? They are like a network of tubes, right? Then what it is, this red color one is a mitochondria. This is another mitochondria. All right. If you can cut open inside it, you can see it has inside the membrane system as well. It's a mitochondria. It is involved in what? Does anybody know? It is the powerhouse of the cell, right? So it is involved in production of energy, right? This I'll tell you. This is called a centriole that is involved in cell division, right? Forget about that for a minute. This is what you call as the Golgi complex or Golgi apparatus. That is sort of like a let's say like a central post office. It takes packages and sort of repackages them and delivers them to a different place. I think you have this in O levels as well. So this is a typical animal cell. All right. We'll go to a plant cell. So the biggest difference of a plant cell, Putha, is other than this outer covering, that is your plasma membrane, the usually plant cells have a much more thick and much more, let's say, firm, stronger structure called the cell wall. Can you see? It's the thick structure called the cell wall. Cell wall. If you remove this cell wall, you can see it has a nice plasma membrane, and I'll remove the plasma membrane as well. This is your jelly-like cytosol. Jelly-like cytosol that is inside. And let's remove that as well, cytosol. Now all the cellular components are floating, right? So the plant cells have a sort of a very big structure that is not the nucleus. So their nucleus is somewhat pushed to a side. And usually plant cells have this big structure called a central vacuole. A central vacuole. Right? It is just, let's imagine it's a big water tank. Right? It's a big water tank containing mainly water and some dissolved things such as sugars and some colored pigments and different things like that. Right? Central vacuole is sort of the biggest thing that you can see when you observe a plant cell. Right? They also have a nucleus that is sort of pushed to a side. Right? Pushed to a side. Inside here you have the nucleus, so you have this smooth endoplasmic reticulum, right? And the rough endoplasmic reticulum. And inside that you can see the nucleus, right? It is a bit covered. Okay. Then RNAs. The other special feature is this. These green color things are what? The chloroplast that is involved in photosynthesis. Inside it, you have this small, let's say, what we call as bags of membrane, right? They have been proper lasant arranged, right? So this is a very, a special feature of a plant cell. Not only plant cells, any organism that can photosynthesize as this organelle, that is your chloroplast. So that is just the basic tour. I'll show you a bacterial cell as well. A bacterial cell, or what we call as a prokaryotic cell, right? It's not a properly organized cell. It's like someone who has it's not a properly organized cell. So it just all the functions, but not in a very organized way, right? It's not properly organized. So there's no especially nucleus, there's no special mitochondria, right? There's no particular endoplasmic reticulum, right? So it's a bit messy, right? Not properly organized. So that's why we call it as prokaryotic, not properly organized. Okay, so he doesn't have a nucleus. So his genetic material, the DNA, nucleic acid, is sort of random. Just lying there. And they usually have outside their cell membrane, they can have a capsule that is similar to a cell wall, but a bit different, right? So we also call it as a cell wall, right? We also give it the special name as a capsule. So I'll tell you something interesting. So if you want to kill bacteria, what do you drink? if you have a bacterial infection. So I might have to drink this if my this the cold that I'm having doesn't go away. What do you drink when you have a bacterial infection? An antibiotic, right? Antibiotic. So when we are prescribing antibiotics, we have to consider. So there are signs. So when you have been infected by a staphylococcus or a streptococcus, there are so many bacteria. So when you have been infected by a bacteria, you have to think: does this bacteria has this capsule? Capsule. So if they have a capsule, what is it going to do? That bacteria is harder to kill. Antibiotics, it's harder to kill that bacteria. So when they are designing antibiotics, when they are discovering antibiotics, when they are sort of when they are creating, as well as when we have to prescribe someone antibiotics, we have to think about the qualities of the bacteria. Are they having a capsule? So these capsulated bacteria are sort of harder to kill. Anyway, so inside the capsule they have a normal plasma membrane. They have this cytosol as well, right? And they can have this big tail-like structure that we call as a flagellum that gives them the ability to move. A flagellum, right? So that is basically bacterial cells. So these are the three main, let's say, the designs of cells that you can need to know. So there's a small video. I don't know if there's.
Cells are the basic living units of tissue.
Can you hear the audio?
Cells are the.
Is it laggy? I have paused it. Just let's give it a try. Just an introductory.
Basic living units of tissue. The human body is composed of trillions of cells. There are over 200 different cell types. Each specializes to perform essential functions. Bone cells form and regenerate the bones in the body. Nervous system cells send messages within the brain and throughout the body to produce actions. Cells in the blood carry oxygen and help the immune system. Male and female sex cells unite to produce offspring. The genetic information inside each type of cell acts as an instruction manual telling a cell how to function and replicate. Cells are the basic living units of tissue. The human body is composed of trillions of cells. There are over 200 different cell types. Each specializes to perform essential functions. Bone cells form and regenerate the bones in the body. Nervous system cells send messages within the brain and throughout the body to produce actions. Cells in the blood carry oxygen and help the immune system. Male and female sex cells unite to produce offspring. The genetic information inside each type of cell acts as an instruction manual telling a cell how to function and replicate.
Right, okay. So there's a question asking how do we know that the antibiotics are not, antibiotics are only killing this sort of harmful bacteria, not your body cells. So they are designed to target structures only bacteria have. So when we are discussing this chart, when we'll talk about different I mean, not here exactly, but when we are doing evolution, that is your third lesson, we'll go into much detail about the sort of the structure of bacteria. So your antibiotics are designed to sort of attack only structures that bacterial cells have, right? Bacterial cells have, particularly in their cell wall, they have special proteins that these antibiotics will go and dissolve. Then what is going to happen? Water is going to go inside the cell and the bacterial cell will burst. That is one mechanism, right? We call them as cephalosporins because they make a pore and water will go inside and the bacteria cell will, right? It will, let's say, explode. So that's just an example, right? Yeah. Please send messages to the main chat, Puta. Okay. So I have to slide and see, and your messages will be kind of ignored if you send them to me privately. Okay. So let's go into the first structure that we have to discuss. That is the plasma membrane or the cell membrane. It is very, very important because the cell is surrounded by this plasma membrane or the cell membrane. It is the outer boundary, outer limit. So, two people not discovered, they actually described, right? In your resource, page 27, Puta, right? Singh and Nicholson, they put forward what we call as the fluid mosaic model. Okay. Fluid mosaic model. That basically says that your cells have two natures. That is, it has a fluid, sorry, the cell membrane has two, let's say, characteristics. It has a fluid nature and it has a mosaic nature. I'll tell you what are these. Fluid mosaic model. Right, fluid mosaic model. So you should be able to draw this, but I have given you a diagram. And the end of the tube, there's some space at the end of the structure essay questions, and we are going to write some essays. So in that essay, I want you to be able to draw. So let's just see what they're saying about the plasma membrane in the resource, right? Mainly, it is composed of two things. Phospholipids. I hope you remember the phospholipid structure. What does it look like, a phospholipid? How do we simply draw a phospholipid? It has a, I hope you have studied, it has a hydrophilic head. The head likes water. And it has two hydrocarbon tails that is composed of your what? The fatty acids. So the head is Puta hydrophilic. If you don't remember today itself, you will have to go back and read about your phospholipids. And it has two hydrophobic tails. Because why? The hydrocarbon chains of the two fatty acids don't like water, no? But the head is composed of your glycerol and your some polar molecule, as well as we say even in the fatty acid, the corner carboxylic acid part it doesn't hate water. So, right? So the hydrophilic head likes water and the hydrophobic tails don't like water. So remember that when we are thinking about the phospholipids in this plasma membrane. Okay. Then proteins, we'll talk about how these proteins are arranged. Okay. Right. Then what does it say? Plasma membrane has the following features. It is about 7 nanometers thick. So imagine how thin it is. Right? It is about 7 nanometers thick. What is the thickness? 7 nanometers. And mainly made up of a phospholipid bilayer. Phospholipid bilayer. Okay. I'll draw the phospholipid bilayer first. So this plasma membrane is actually one of the basic things that you should be able to draw even when you go to your university, right? If so, you're doing bio, no? So if you choose to do something bio related, even if you go to even if you go to a medical faculty, I remember So we have a subject called biochemistry in the first 2 years. So we have a sort of a session called a tutorial, right? A tutorial we have to compulsorily attend. Danimaring Andrew. Lectures are not compulsory in medicine. So you don't need to go to lectures, but you have to go to these tutorials definitely. And one of the first tutorials that we had for biochemistry is actually cellular structure, right? Cellular structure. And so there's a lecture in every tube, there's a lecturer, and he's going to ask you questions. And he's going to, they are giving you questions beforehand, and also they are going to sort of give you questions right there. You will have a question at the, you are expected to write the answer. So imagine this is the sort of the first week of proper studying in the medical faculty. So we are very, very nervous. And one of the first things they asked me to, he stood me up and he asked me to come to the board that is in front of like 30 people. And he asked me to draw this plasma membrane, the fluid mosaic model, right? So even if you go to medicine, you cannot run away from these basic things because it is so important. Okay. So let's draw this. What does the first statement say? It is mainly made up of phospholipid bilayer. What do you mean by a bilayer, Buddha? It means I'll draw phospholipids like this point onwards. I'll draw the head and two tails, head and two tails, head, two tails, head, two tails. Okay. And what do you mean by a bilayer? That means it has two layers. Right? So one layer is this. I'll draw one more phospholipid for your benefit. Then the other layer is like this. Upside down. Two layers. This is a bilayer. Why? Why are these phospholipids arranged like this? I hope you can see. Why are these phospholipids arranged like this? Because Putha, inside the cell as well as outside the cell, outside the cell, you have water. What do you have? You have water. So what did I tell you about these heads and tails of the phospholipids? The heads like water and the tails hate water. So can the tails live with water inside and outside? So imagine this is the inside the cell, inside and outside. So I now remember. So the lecturer blamed me, sort of scolded me for not labeling which side is the inside, which side is the outside. He sort of demanded. So this is the inside and this is the outside, right? So the heads like water and the tails hate water. So what is it going to do? So the tails have to be protected from the water. How do they do that? The tails are oriented inside to the interior and they are covered from the water that is outside and inside by the heads. So the heads are facing the water and the tails are shielded from the water by the heads. So this is what the this is the phospholipid bilayer. Phospholipid bilayer. Okay. Any questions? Then they say phospholipids are amphipathic molecules. What are amphipathic? They have both hydrophobic, hydrophilic natures in the same molecule. I hope you remember. Right? That's what we know. The hydrophilic head of the phospholipids face outwards to the aqueous environment inside and outside. So that's what I just told. The heads are facing inside and outside. Got it? The hydrophobic tails face inwards and create a hydrophobic interior. So this interior of the plasma membrane, that is the middle of the plasma membrane, is a protected place for this hydrophobic tails. Got it? And now we come to this fluid part of the model that they are using to describe this plasma membrane that they are using to describe fluid. Fluid means that it's like a liquid, no? It is, that means it's not very stiff. It is able to sort of move freely. That is because these phospholipids are, as you can see, they are not connected by anything, right? They are not connected by anything. They are just oriented freely, right? Imagine like something. It's like a series of, like this. It's like foam floating on the ocean. Like they are not, they are freely moving with the ocean waves, right? A layer of foam. Have you seen like penalty in the movie, right? So when the ocean waves are moving and they can sort of freely move. It's not like a very stiff covering, right? So that is why, because we call this plasma membrane as fluid. It has fluidic nature. Then there are proteins, right? This is so important. The proteins, there are so many proteins that are embedded in the plasma membrane, right? I'll draw another phospholipid here. There are several types of proteins, right? So I'll just complete this diagram and explain to you, right? So I'll draw a big protein like this. That is sort of spanning what we call as the entire thickness of the plasma membrane. There are proteins. Okay. Then I'll draw some more phospholipids. Bit longer, no? The tails are getting short. Then there are some proteins that are not that tall. It's short like this, right? But they are anyway embedded in this bilayer. They are not tall, but they are anyway embedded, right? So, right. What else? So proteins molecules embedded randomly contribute to its mosaic nature. Okay, give me 1 minute. So if you go to your tube, I have given you a good diagram of the plasma membrane. I'll explain. Oh, wait, not yet. I'll explain this bit later. Okay, I'll explain this bit later. Okay, just follow me here, right? Then I'll draw another protein here that is also sort of like this, that is spanning the entire thickness, right? Then I'll draw more phospholipids like this. This is basically it. And yeah, I'll draw a bit more and draw another type of protein. There are different types of proteins, okay? And thus I'll draw another protein here that is called a peripheral. Okay, so this is what you are supposed to draw. Then additional components I'll draw now. So connected to these proteins, there can be some antenna-like structures. I'll draw them simply like this, antenna-like structures, right? And in between these phospholipids, there can be some cholesterol molecules. Cholesterol randomly here and there, some cholesterol molecules. I'll explain to you the function. So this is your fluid mosaic model, right? So I label them. These are both actually these two proteins, what we call as integral proteins. Integral proteins. Integral proteins. But this one, can you see? There's a channel going through it, right? There's a hole through it. Can you see? There's a hole. That means it's like a gate. It's like a gate. So things can come through this what do you call as this protein into the cell, right? So it is what we call as a transmembrane protein. This one is a transmembrane protein. Okay. And this one, this small one, I hope you can see. This is not sort of, let's say, embedded, but it's still a protein. We call it as a peripheral protein. That is sort of just lightly attached to the inside of the plasma membrane. That is called a peripheral protein. And I told you this sort of antenna-like structure that is sort of attached to this protein. We call them as it is a it's mainly a carbohydrate structure. It is called a glycoprotein. 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 Right? Similarly, such carbohydrates can be attached to your this sort of phospholipids as well. Right? Phospholipids as well. They are called as what? So phospholipids are what? They are lipids, no? So they are called glycolipids. Glycolipids. Right? And this one, sorry, this one is also an integral protein. Okay. Right, I'll explain. And then what did I forget? The cholesterol. So this green color is a cholesterol molecule. Cholesterol molecule. Right. Can you see the board clearly? The diagram, is it the clear, right? There's a lighting issue. That's the next thing I have to improve. I'm going to install some lighting to the board so you can see better. So this diagram you have to be able to draw. Okay. This diagram you have to be able to draw. So what did I have? Why do I have to explain? Hold on. So yes, we have integral proteins and peripheral proteins. And then we have cholesterol, yes, we have glycoproteins and glycolipids, yes. That's basically it. Now I will start explaining, okay? Right. So Buddha, when talking about the proteins, there are two main types. There are integral proteins and peripheral proteins. These are the two main types of proteins. All right. All proteins are either integral or peripheral. Integral means they are anyhow embedded inside the the phospholipid bilayer. Embedded inside. So you can see this one is slightly, only about halfway. This one is even a bit taller than that. This one is going the entire full thickness, right? So anyhow, no matter how tall they are all called integral proteins. But like you can see, this peripheral protein is not actually inside, but it's just stuck to the bilayer, phospholipid bilayer. So therefore, it is called as a peripheral protein. But when we are thinking of the integral proteins, there's a special type of integral protein called the transmembrane protein. That is, I told you, it is the entire thickness. It's a special type of integral protein. And usually, Buddha, these transmembrane proteins have this channel inside, right? As I told you, it can act as a sort of a gate for substances to come in and also substances to go out. Right? That's a transmembrane protein. So your resource say some of the integral proteins penetrate all the way through the membrane called transmembrane proteins. So these are transmembrane proteins. And some others penetrate only part of the way into the membrane. Then they say in the page 28, most of the integral proteins are transmembrane proteins which have a hydrophilic channels. Right? So I'll add a new label. So what is this, Mutade? This channel, hydrophilic channel. Hydrophilic channel. Right. Some proteins are not embedded in the bilayer at all, but are loosely bound to the surface of the membrane called peripheral proteins. This one. Got it? Those are peripheral proteins. So they are not embedded. Some proteins and lipids have branching carbohydrate chains like antenna, forming your glycoproteins and glycolipids. Animal cell membranes may contain a few cholesterol molecules. So I drew the cholesterol molecules randomly integrated into the bilayer. So Muta, this cholesterol sort of maintain a balance, right? Because we'll talk about this especially in when you're talking about environmental conditions. Different temperatures can have effects on this fluid nature. So I told you this phospholipids have a fluid nature, right? But when the temperature actually decreases, what do you imagine? Their fluidity somewhat decreases because they start to sort of get close to getting frozen, right? Get close to getting frozen when the temperature drops. So these cholesterol are not very easy to freeze because they are steroids, so they have a complex structure. But because these phospholipids are sort of very simple, you can sort of lock them into a crystalline structure because they have a simple. They are like have you seen Legos? You can like lock them into place. No? But the phospholipids are easy to lock into place. But having these random cholesterol molecules here and there, it's going to make that difficult, right? So when the temperature drops, it maintains membrane fluidity. Such an important feature. The cholesterol prevents the membrane from freezing. Okay. So they provide rigidity and stability. Also, when the temperature is very high, the opposite happens. The phospholipids tend to sort of move away from each other. Because the temperature is high, they get some of the thermal energy. They become very sort of, they start vibrating sort of. They start to sort of drift apart. That is bad as well. So the cholesterol act as sort of like a glue between the phospholipids to keep them from moving apart. So it performs two things. So it maintains the fluidity when the temperature is low, and it gives stability at sort of high and at the normal temperature. You understand? Any questions? Specific number to draw those. What draw what, Putha? Specific number to draw what, Putha? Right. So let's go to the diagram that I have given in your tutorial, and let's label it. Okay. Right. So I think this is a bit too detailed for you, but can you see? This is a transmembrane protein. Why do we call this one a transmembrane protein? Because as you can see, it covers the entire thickness, right? It covers the entire thickness. So this is your phospholipid bilayer, Putha. Can you see? These are the hydrophilic heads. These are the hydrophilic heads. And this is what? The hydrophobic interior. So this is a transmembrane protein as well. But the difference between them is what? This has a channel. This does not. This is a peripheral protein. This is also a peripheral protein. You can see my screen, right? Yeah. This is also a peripheral protein. Okay, so what is attached to this? This is a protein. So this is a glycoprotein, right? This is a glycoprotein, and you can see some of these carbohydrates attached to lipids as well, phospholipids. So those are glycolipids. Okay, that is basically it. All right. Can you see? In between your phospholipids, you can have cholesterols. You can have cholesterol. All right. Okay, let's write down a small note here. All right. Uh, manage your space, Putha. Okay. Manage your space. Right. Write down. Singer Nicholson. Singer and Nicholson. Singer and Nicholson. Singer and Nicholson introduced the fluid mosaic model. Singer and Nicholson introduced the fluid mosaic model. Singer and Nicholson introduced the fluid mosaic model. Fluid, right? Fluidity, right? Like this fluidity. Fluidity given by what? The phospholipids, right? Fluidity, and draw an arrow, phospholipids. Fluidity given by phospholipids. Then write fluidity arrow given by phospholipids. Then the mosaic nature, underneath that mosaic nature, by proteins embed, draw an arrow, mosaic nature, draw an arrow, proteins embedded randomly. Proteins embedded randomly. Okay. All right. Then write down. There are two main types of proteins. There are two main types of proteins. There are two main types of proteins. Put number 1 and 2. Integral proteins and peripheral proteins. Integral proteins and peripheral proteins. Put a star mark. Put a star mark. Put a star mark and write transmembrane proteins. Transmembrane proteins. Transmembrane proteins. Transmembrane proteins. Like in front of it, right? I draw a small line and write special integral proteins. Special integral proteins. Special integral proteins that penetrate. Special integral proteins that penetrate. Special integral proteins that penetrate all the way through the membrane. Transmembrane proteins. Okay. Then write down underneath there, star mark again, importance of cholesterol. Importance of cholesterol. Importance of cholesterol. Importance of cholesterol, like underline it and write down under that. Importance of cholesterol. And any questions? Did you miss anything? Anyone? If you missed anything, please repeat the last. Which last one, Putha? Which last part? Yeah. Transmembrane proteins. There are special integral proteins that, what did I say? Can someone put in the chat? Special integral proteins that penetrate all the way through the membrane. Transmembrane proteins. Then okay, so we were talking about the importance of cholesterol, right? Importance of cholesterol. Write down. Cholesterol maintains fluidity. Cholesterol maintains fluidity at low temperatures. Cholesterol maintains fluidity at low temperatures. Maintains fluidity at low temperatures. And gives stability and give stability at moderate temperatures. At moderate temperature. Right. I think that is enough about the structure. What is more important is the functions, right? The functions. So, number one, you have to be able to, so you have 6 functions in your resource. You have to be able to write down all 6, right? Right now, get the basic idea, right? Understand each function, then you can focus on memorizing, right? As I always say, without understanding, there's no memorization, right? You cannot, I mean, there can be, but those are, I mean, it's not a human way or a smart way to learn, right? Understand something and then memorize. Then it will go to automatically to your memory because you understand it to the level that you can teach it to someone, right? So when you are studying something, think, do I know this enough? If someone asks me to teach this to them, can I teach it, right? So if you understand something enough, you have to be able to teach some confident. So always think before moving on to a different topic, even in physics or chemistry or wherever. Think, have I understood this topic to a certain degree that I am able to explain this to someone else? So that is the level of understanding you should have, right? Because A level science is not your, I mean, Sinhala literature, remembering your all these kavi. No, it's not like that, or history, remembering all this LDP or whatever. Be logical about it, right? So, okay, first function, that is sort of the basic thing that we can think of, right? Because the plasma membrane is the outer covering. It sets the limit of the cell. So it is like the wall of a city, right? So it's around the cytoplasm. So it's a physical barrier, right? So inside the cell is a different environment, outside the cell is a different environment. So those two environments are separated by what barrier? Yeah, plasma membrane. That is the first function. Physically separates the intracellular components from the extracellular environment. Okay, physically separates. Right. Then the second one, Uta, selectively permeable and able to regulate the exchange of material needed for survival. Selectively permeable. What is permeability? Right? If something is permeable, that means things are able to cross it. Right? Substances are able to cross. A permeable membrane allows substances to cross. Right? Impermeable means nothing is getting across. But what's the word we use here? Semi-permeable. Right? There are some things that are allowed. There are some things that are not. Imagine it's like a toll gate at the highway. So tractors, motorbikes, people on foot, they are not allowed to cross. But normal cars, buses, vans, everything that is sort of qualified to go in the highway are allowed. So imagine like the toll gate at the highway. So your plasma membrane is like that. Particularly who? These transmembrane proteins with channels. They are these sort of like the gateways, toll gates into the cell and also going out of the cell. Right? Selectively permeable. There are different types of transporters, Putha, right? So there are, I mean, there can be iron transporters, right? There can be just things that can freely come across, right? So things like that. So it's a gate, but you don't need to pay anything. You don't need to pay a fee to come into the cell or go outside. Then there are gates that you need energy. You need to give a fee. Like into the highway, you have to pay and come into the cell. Right? Different types of transporters are there. Okay. What else? Transport. I don't think we need to go into that much detail. After discussion, I'll let's see. Right. Then third one, proteins embedded in the plasma membrane identify the cell, enabling nearby cells to communicate with each other, involved in cell recognition. Particularly, these glycoproteins, together with the protein, sort of acts as like the name tag of a cell. So if you're wearing like a name tag, that means like a nearby cell or a nearby, usually nearby cells can identify, this is this kind of a cell. For example, a red blood cell can have a special type of glycoproteins that are like unique to it, right? Red blood cells can have their own sort of glycoproteins that are sort of like their name tag. I am a red blood cell, right? So if it comes across like, let's say, like a sort of like a defensive cell, such as a white blood cell, then it can say, see, I'm a red blood cell, I'm not harmful, so I'm okay. So it's like that. It's protected and it is identified, right? It's like that. Understood? Uh, okay. Then I'll explain this a bit further, but let's just go through them. Some proteins act as receptors for interacting with specific biochemicals such as hormones, neurotransmitters, and immune proteins. So, Buddha, in the outside of your cell, there are several kinds of messengers and different, let's say, substances that are sort of interacting with this cell. If you take our body, there are hormones, right? There are hormones, right? For example, take a hormone like testosterone, for example, your male hormone. Think that is this plasma membrane belongs to a muscle cell. This plasma membrane belongs to a muscle cell. So, do you know what is the effect of testosterone on muscle? In males, testosterone can be found in females as well. Do you know what effect does this male hormone have on muscle? If you have a lot of testosterone, what do you think? So this testosterone that is coming from outside can be a signal for this muscle cell to sort of get bigger, right? Or even it can signal it to divide, right? This male hormone. So that recognition, now there's testosterone coming from outside, is done by proteins as well. I'll explain that with a diagram. Just go through. Some proteins in the cell membrane can attach to cytoskeletal fibers and help to maintain the shape of the cell. So we'll talk about this. So the inside of the cell, we have sort of like a what do you call that, scaffolding, right? When you are building something, that is sort of an internal supportive system. So you have this sort of tubes and supporting structures that are sort of attached to this plasma membrane. That is inside. It's like a network of tubes, right? That are sort of giving support to the interior of the cell. That we call as the cytoskeleton. Cytoskeleton. It's like a skeleton of the cell, right? It's like a palangi, all right, like a scaffolding that is providing support. So the cytoskeleton can be attached to these peripheral proteins, particularly. It can be attached to these kind of proteins as well, these kind of integral proteins as well. But their purpose is to give support. So maintain the shape is again another function of the plasma membrane. Then some proteins in the membrane act as enzymes, right? Enzymes. So I'll draw this sort of a protein here. It can have this sort of a site like this where different molecules can bind to it and become different, right? For example, if a disaccharide binds here, a disaccharide, this protein can help to break this bond for them to become a monosaccharide, right? That is what we call as a enzymatic activity. Okay, right. So now I'll show you some examples. Any questions with that? I know this is a bit of a lot of information, right? Any questions? Right. So I have diagrams for each of these. So transport protein looks like this. You can take a screenshot if you want. I haven't put. Right? Can you see? There are molecules outside and they can come into the cell through this transmembrane protein with a channel. Right? You can take a screenshot. I'll take one as well. So now it's easier for me to show. So that is one function, no? That is selectively permeable. Everything is not able to come. Then what else? Enzymatic activity. That is the last one. Putha. Some proteins in the membrane act as enzymes. So what do they say? Microvillus on epithelial cell lining of some part of the gut contain digestive enzymes. So imagine this is a cell in your digestive system. So here is downward is a cell, no? From here downward it's a big cell, right? Imagine it's like this. So this is inside, Putha. In this diagram, this is inside, this is outside. So imagine this is your digestive system. So this tube outside is your digestive system. So you have eaten something. For example, my example, you have eaten a disaccharide, you have eaten sucrose. Right? So the sucrose have to be broken down into monosaccharides to be used as energy into glucose and fructose. So the digestive system cell can have this sort of a protein that can act as enzyme. I told you enzymes are mostly proteins. I told you, right? Anybody who remembers? Yeah, enzymes are mostly proteins. I hope you remember. Yeah, I'll go through the functions. Just a sort of a brain teaser. What structure of proteins are acting as enzymes? Is it primary, secondary, tertiary, or quaternary? What structure of proteins? What level of protein organization acts as enzymes? Tertiary, right? Tertiary structure. Okay, right. So this is such an example of the enzymatic. Then what is this? Yes, cell recognition, cell to cell recognition. Can you see? So I told you there can be glycoproteins or even glycolipids attached to your phospholipids. They can have that sort of acting as a name tag of the cell, right? I am this cell, I am good, or I am bad. They both could be indicated, right? You don't need it. Yes. Attachment to cytoskeleton. Can you see? So this is the inside of the cell now. This is the inside, this is the outside. So these are part of that skeleton of the cell. That is sort of like an internal network of supports that is providing, like maintaining the shape of the cell. attachment to cytoskeleton. Yes, that is all that you need to know. Any questions? Let's move on to do some MCQs. This question, Buddha. This MCQ, I need the answers in 2 minutes. Please do. Right. Okay. I'm getting answers. Let's see. What does the question say? So there's about 10 people. Where are the answers? Other people. What about others? Right. Let's discuss which of the following statements regarding plasma membrane is correct. It's mainly composed of carbohydrates, phospholipids, and proteins. Okay, what is wrong about that, Puta? So I gave you the answer. I mean, it's wrong. So what is wrong about that? So I want you to write a review in front of it. Right? So there's nothing to review here. It is mainly composed of what? Phospholipids and proteins only. There is no main carbohydrate composition. Right? Phospholipids are movable and provide a fluid nature. Right. So that is sounds okay. That sounds okay, right? So for now, it is a good answer. Peripheral proteins are tightly attached. No, it says your peripheral proteins. What is the definition? Not embedded in the lipid bilayer at all, but are loosely bound to the surface. Those are peripheral proteins. So what is wrong? Not tightly, loosely. Phospholipid bilayer enables nearby cells to communicate with each other. Is it the phospholipid bilayer? Nearby cells to communicate. Is it the phospholipid bilayer? No, right? It is the proteins embedded. Right? It is the proteins embedded. So write a review. Proteins embedded in the plasma membrane identify the cell. Proteins embedded in the plasma membrane identify the cell. Proteins embedded in the plasma membrane identify the cell. Within brackets, cell recognition. Within brackets, cell recognition. Then hydrophobic tails of phospholipids attach to cytoskeletal fibers and help to maintain stability to that. So it's missing, okay? So it is missing a word. I apologize. Just be stability, okay? Stability. Right. So, okay, cytoskeletal fibers have a role in stability, but does it have to do with anything with hydrophobic tails? No. Right. What do they attach to? Usually, are integral proteins. Right. Integral, or it can be peripheral proteins as well. Give me 1 minute. Right. It can be peripheral proteins as well. Anyhow, cytoskeletal elements and your phospholipids have no interaction at all. So it's a good question, right? Okay. So now please write down your homework for this week, right? Write down your homework for this week. It's an essay. It's an essay. Describe the fluid mosaic model of plasma membrane. So that is 1A. B, describe the functions of plasma membrane. So this is going to be your homework, and I'm going to give you a hint, right? So in the paper, they won't tell you that if you want to draw that you are required to draw a diagram or not. They won't tell you that. right? They won't tell you that you are supposed to draw a diagram, but they will give marks. So if you haven't drawn a diagram, you're going to lose marks. Clear? So at the end of your sort of, not at the end, by the time you are in your grade 13 or in your final year, you have to be able to sort of, let's say, have a guess, like thinking this topic requires a diagram. That comes with your expertise in the papers that you have done past papers, as well as you have to be sort of familiar with the resource. This topic had a diagram, so I have to be able to draw that diagram, because that will get me marks. So because you are very early right now, I'll tell you. So when you are given such a topic, describe the fluid mosaic model, you are supposed to draw the fluid mosaic model, right? You are supposed to draw. So start this essay right now. Draw this diagram. Draw this diagram. That is the beginning of this essay. Take 10 minutes and draw this diagram. I will stop screen share. You can see, right? You can see clear, Yep. Okay, there are some questions. Can ordering the proteins in plasmid chain how immune cells recognize it? Yeah, that's basically one of the methods how chemotherapy works. But in chemotherapy, one of the risks is it's not very precise. So when you are having sort of killing this sort of deformed cells, they don't usually have any sort of special recognition per se, right? So it's very difficult for the chemotherapy to actually identify your own, proper cells and deformed cells. So it is a big difficulty, right? So when you are for example, when you are giving chemotherapy, we can only target particular characteristics. So, a cancer is like a group of quickly dividing cells. Quickly dividing cells. We are quickly dividing. So, a particular mechanism of chemotherapy is you can target quickly dividing cells in the body, right? Quickly dividing cells. So, when you're targeting quickly dividing cells, other healthy cells that are quickly dividing will be affected. Like what? Your hair follicles, they have quickly dividing nature. They are quickly producing hair, so your hair can fall out. In your digestive system, your GIT system, the interior or the inner lining of your digestive system has quickly dividing cells. So they can be targeted by chemotherapy as well. So your digestion can be affected. So chemotherapy has so many side effects. And yes, so cell recognition can be applied in proper chemotherapy, but still in development, I think. So cholesterol does not provide fluidity, it maintains fluidity. Fluidity is by the phospholipids, right? Your cholesterol has sort of, as I told you, like if your phospholipids are sort of building blocks, your cholesterol can come in between them and prevent them from interlocking with each other, right? When the cholesterol amount in the body is high, what happens? High cholesterol. High cholesterol has to do with the condition that we call as atherosclerosis, right? Atherosclerosis. So that is not related to your cell membrane. So you have your blood vessels, no? You have your blood vessels. High cholesterol can lead to cholesterol deposition on the inside of your blood vessels like this. like a lump of fat. So this can sort of impair the blood flow. The blood flow will be very disturbed. So this can lead to other conditions. So if this blood vessel is in the heart, your heart is not getting enough blood, it can lead to death of heart muscle. Right? But here we are talking about the benefit of cholesterol. You have to have cholesterol because I told you, cholesterol is the source for your what? Steroid hormones, I told you. There are hormones called steroid hormones. Those are synthesized from cholesterol. Your steroids. So your steroid hormones are necessary. Your testosterone, estrogen. These are steroid hormones. Without them, you cannot have a good. I mean, the body cannot function properly. Okay. All right. Okay. Drawing. Okay. 5 more minutes. So tell me, is this camera arrangement a bit clearer? The body is much clearer, right? You can see the board, right? Yeah. Okay. Okay. I think we can finish the class a bit early, no? So because the next structure explain the fourth function. What is 1, 2, 3, 4? Act as receptor molecules, such as hormones, neurotransmitter, and immune proteins. So this is again a bit like cell recognition. Okay, I'll give you an example. Hold on. So another example that I can give you is imagine. So usually a hormone can bind to a receptor and give a sort of change the way the cell behaves. For example, imagine that hormone is insulin. You know, insulin is a very common hormone, right? So insulin, when binds to the cell membrane, it can stimulate the transport of glucose into the cell. Glucose is going to come in. So imagine like insulin is like the key to unlocking this gate, unlocking this door, right? So there can be so much glucose outside, but without the key, that is the hormone insulin, the glucose cannot get inside the cell. So when we are talking about diabetes especially, we say starvation amongst plenty, right? කෑම ගොඩක් තියෙද්දි බඩගින්න ඉන්නවා. That is because when you are diabetic, you have so much glucose outside the cell. But that glucose can't come into the cell because you don't have the key. What is the key? Insulin. Right? So much glucose is outside the cells, that is in the blood circulatory system. So that is going to cause so many damages. Right? Have you seen do you know what are the damages of too much glucose? That is the damage of diabetes. It can cause eye damage called retinopathy. It can cause kidney damage called nephropathy. Right? It can cause small blood vessel damages that can cause wounds. Right? කකුලේ තුවාල, diabetic ulcers කියලා අහලා තියෙනවද? Do you have any old grandparent with diabetes? Right? They can have non-healing wounds. right? So that's the example. So insulin can change some protein, so they allow the transport of some substances. So the previous example I gave you, if the hormone is testosterone, it can change the cell. Maybe the cell can grow in size. If it's a muscle cell, when you give it testosterone, it can increase in size. Understand? No need to think about that in much detail, right? So as I was saying, next we have to talk about the nucleus, right? Nucleus is, as I told you, it is the most significant organelle in an animal cell as well as in any cell, because nucleus is sort of like the main control center. Main control center. Without a nucleus, it's like a plane without a pilot. There's no purpose, there's no control. All right. So nucleus we have to talk a lot in detail. So maybe we can start that topic next week, right? And so if you have completed drawing this, hands up if you're still drawing. No one is still drawing, right? Okay, we can finish a bit early today, no? So because some people don't have the tute as well today, no, so printed, it's my fault.